Imagine holding a massive pile of raw biological code, thousands of A's, C's, T's, and G's. On their own, these sequences are just a scramble, offering no immediate indication of how they relate to one another. To find those relationships, we use multiple sequence alignment. This process arranges sequences side by side to identify regions of similarity, which helps us find evolutionary links and predict potential functions. In the next 6 minutes, we are going to take a messy block of raw data and use Clustal Omega to organize it into a structured, color-coded map and a branching evolutionary tree. You only need 3 things to follow this workflow: your raw sequence data, a basic text editor, and a web browser. Structuring genetic data into an aligned map provides the necessary context for comparative analysis. Without this step, these sequences remain a list of isolated samples rather than a related set of evolutionary data. Step 1 is preparing your data. To ensure the software processes your information correctly, we need to format the raw sequences into the standard FASTA format. The FASTA syntax is straightforward. The first line must start with a greater than symbol, followed by the sequence name. Your actual genetic code begins on the very next line. Before proceeding, scan your file for duplicate sequences. Redundancy clutters your results and increases the processing time without adding new information. Manually delete any copies and ensure there are no blank lines between your different sequence entries. Once the text is clean and continuous, save your document as a simple .txt or .fasta file. Sticking to this precise formatting standard prevents the algorithm from throwing errors when it attempts to parse your data for alignment. Now, navigate to the EMBL EBI Clustal Omega web server. This interface is the most accessible starting point for aligning standard datasets. You can move your formatted FASTA data into the tool by copying and pasting the text directly into the input box or by uploading the saved file. Next, we choose the output format. Options like Clustal, FASTA, or Phylip determine which visualization software you can use for the final analysis. We also need to consider gap penalties. The algorithm introduces empty spaces, or gaps, to bring segments into alignment. High penalties discourage breaking the sequence, while lower penalties allow smaller gaps to force a match. Be aware that the order in which you input your sequences can influence the initial guide tree the tool uses to build the alignment. Submit the query and check the status window to confirm the system has accepted your data without format errors. Configuring these specific parameters ensures that the gaps in the final result represent probable evolutionary events rather than mathematical artifacts of the algorithm. Once the process is complete, download the output and open it in a viewer like JALView or Mega. Your alignment is now visible as a structured, color-coded grid. Here, vertically stacked sequences with matching data share the same background colors. Your first step is to scan the grid for conserved regions. A conserved region is a vertical column where every sequence element matches perfectly. These blocks point to critical biological functions. In contrast, variable regions show high diversity across columns, often indicating regions of rapid adaptation. These variations can also help researchers identify mutations that may be linked to specific diseases. From this alignment, you can also generate a phylogenetic tree. This visualization clusters the most similar sequences together on shared branches to show their ancestral proximity. By identifying these distinct visual patterns, you convert a raw jumble of abstract code into a structured map of our deep evolutionary history. You have now transformed raw, unorganized data into a high-quality multiple sequence alignment. This alignment allows you to compare healthy and diseased sequences to pinpoint mutations or predict the function of unknown proteins based on their similarity to known ones. While the web server is ideal for standard projects, it will struggle or fail when tasked with massive, high-throughput datasets. If you are dealing with thousands of sequences at once, the Clustal Omega command line interface is the required tool for automated batch processing. You can find a link to our tutorial on using the command line version in the description below. Mastering these alignment workflows gives a researcher the foundation to model how protein and nucleotide sequences change and function across different species.